Hello everyone, welcome to the ITLS Academy. In today we are going to discuss our topic that is your microbiology of a cream. Okay, today we are going to discuss about what is the microbiology of the cream. But first of all, we have to discuss about what is this cream. Okay, microbiology is again we all know that the microbiology are what? Microbiology is the study of the microorganism. Study of the microorganism. Now, these microorganisms are either beneficial or either it is harmful. There is only two category of the microorganism. Either they are beneficial or either they are harmful. Beneficial microorganisms are good for the health purpose. Good for the health purpose. But this harmful microorganisms, okay, they will what they do, either they are pathogenic, if you talk about in terms of bacteria, pathogenic, or either it will spoilage. Okay, now this pathogenic bacteria, what they do, they cause a disease. And this spoilage microorganism, they spoil the product and ultimately affect the health of the human being. Ultimately affect the health of the human being. Okay, so in today's lecture, what should we are going to do? We are going to discuss about how this microflora of the cream are. Cream are what? Cream are the white, in a basic definition, white thick portion of the milk, which come in a upper, which form as a upper layer. Okay, so here what should we are going to do? We are going to discuss about what is the microflora of that cream. Okay, so let's start today's lecture. Now, the introduction part. First of all, we are going to discuss what is this cream. So we all know that cream is what? Cream is a fat-rich portion of the milk which is obtained by when we put a milk in a standby condition without disturbing it, and either with the help of the centrifugation. Okay, and this milk, this cream produce around 3.4% of the total milk production. Okay, and this cream are commonly deemed in different ways, um, uh, like that portion of the milk which is rich in milk fat, part of the milk rich in fat which has been separated by the skimming or otherwise that portion of the milk into has been gathered and which contain a large portion of the fat. This is your cream. Now see what is written in a slide. Cream is the fat rich portion of a milk obtained on keeping it undisturbed for some time, gravity separation, or on a centrifuging. However, the figure for a cream production declined to 3.4% of the total milk production. Cream has been commonly deemed in different ways, like that portion of the milk which is rich in milk fat, that part of the milk rich in fat which has been separated by skimming, or otherwise that portion of the milk into which has been gathered and which contain a large portion of the milk fat. Now the next is your, when milk fat is concentrated into a fraction of the original milk, that portion is known as a cream. Okay, when a milk is concentrated into a fraction of the original milk. Okay, suppose that we have a original milk, but that portion of the milk which have a concentrate, then it is known as a cream. Now, according to our PFA rules 1955, they define a cream as a sterilized cream is product of cow or buffalo milk or a combination thereof which contain not less than 25% of the milk. Okay, so according to this PFA rule 1955, this milk is what? Milk is a sterilized product. And sterilized product means that it is free from any type of microorganism, free from any type of microorganisms. Okay, and it is either obtained from the cow milk or either obtained from the buffalo or the combination of both. Okay, and it will contain not 25% of the milk fat. So now we are going to discuss about the types of the cream. So on the basis of uses, this creams are divided into three part. That is, sorry, this cream are divided into several part. Okay, like first one is your market cream, then manufacturing cream, and baker's cream. Market creams which we directly consume. Okay, the cream that we directly consume are come under the category of this market cream. Next is your manufacturing creams. This manufacturing creams is diverted to a manufacture of milk product like butter, ghee, ice cream, various dairy, dessert, etc. Okay, so this manufacturing cream are used in the production of different product like your butter, ghee, ice cream, and various dairy dessert. In India, cream is commonly used for butter and ghee manufacturing. Baker's cream, the cream which are used in a bakery. Okay, and for the production of mainly a bakery product, then this will come under the category of baker's cream. It usually contain around 30 to 40% of the fat. Okay, and the microbiology of this with this product are more since it is a subjected to a more unhygienic practices. Now, the baker's cream are defined as a cream used by our bakers and others in the food industry is commonly called a baker's cream. It is usually contain 30 to 40% fat. The microbiological problem with this product are more since it is subjected to a more unhygienic practices. Okay, if there is a unhygienic practices, then there is a chances of the microbiological problem because when the in a unhygienic conditions, the growth of the microorganisms will increases. Now the next is your on the basis of fat content, this cream are divided into a six category. The first one is your table cream, then light cream, then coffee cream. Coffee cream is contain around 20 to 25% of milk fat. Wiping cream, heavy cream, which contain 30 to 40% of milk fat. Okay, and the last is your plastic cream, which contain a 65 to 85% of milk fat. Okay, now in UK, 8 type of creams are defined only in respect of their fat content are produced. Okay, now on that basis, there are 8 type of fat. One is your half cream, which contain around 12% fat. Sterilized half cream, again it contain 12% of fat. Single cream or cream, which contain around 18% of fat. Sterilized or canned cream, which contain around 23% of fat. Whipping cream, which contain 35%. Okay, and whipped cream, which contain around 35% fat. Double cream, which contain 48% fat, and clotted cream, which contain around 55% of fat. Now on the basis of processing, the cream are divided into 10 category. The first one is your untreated cream, untreated, pasteurized, sterilized or canned cream, UHT cream, frozen cream, sour cream, clotted cream, cream powder, artificial or reconstituted cream, and imitation or synthetic cream. Okay, now the first one is your untreated cream. Untreated cream are defined as a cream which is not processed or cream which is obtained from a unprocessed milk. Okay, untreated cream means simply that it is obtained from that milk which are totally unprocessed. Okay, and this cream will also not a processed. Now the next is your pasteurized cream. Pasteurized cream are defined as a cream which is obtained from a pasteurized milk. Okay, which means that when we pasteurize the milk, then after that the cream which we obtain is called a pasteurized milk. The next is your sterilized or a canned cream. This cream is obtained after sterilization. UHT cream, cream which has been subjected to a UHT processing. Okay, so UHT cream are those cream which in which we have performed the UHT. Frozen cream, the cream which are frozen below a minus 10 degrees Celsius. Sour cream which are obtained by ripening the pasteurized sweet cream to a high acidity, that is around 0.6% of lactic acid. Clotted cream, the cream which is obtained by skimming off the surface layer of a cream which are heated between 70 to 80 degrees Celsius, and then after that it is cooled down in a shallow pan. Now cream powder made by dehydrating fresh cream or mixture of an anhydrous milk fat and skim milk powder. Next is your artificial or reconstitute milk. This type of cream are made up of butter or milk. Okay. The next is your imitation or synthetic cream. This is made by emulsifying edible fat or oil in water with other permeated additives. Okay, now the next is your chemical composition of the cream. We all know that this cream are depend upon totally the composition of the milk. Okay, and it will milk fat is vary from 18 to 85%, and SNF constitute are present in lower proportion. Okay, when a fat concentration is high, then the SNF concentration will be low. SNF means that solid, not fat. Okay, then it should be lower portion in milk. The higher the fat content in a cream, the lower the SNF value. Okay, now see what is written here. In cream, milk fat may vary from 18 to 85%, and the SNF constitute are present in lower proportion than in milk. The higher the fat content in cream, the lower the SNF content. Now this is your constitute. Okay, this is your percentage, 1 or 2. Okay, if fat is 25%, protein 0.2%, lactose 0.3, ash 0.056, solid not fat, that is your 0.680, total fat 31.80, water is 68.20%. Okay, this is what this is type A. Now the next is your, if the fat concentration is 50%, then the protein concentration is around 1.69%, and lactose concentration is around 2.47, ash content is around 0.3, solid not fat content is around 4.5, total solid is around 54.5%, and your water content is around 45.45%. Now the next is your microbiological standard. Okay, now if we're talking about the BIS, okay, the BIS means Bureau of Indian Standard. Bureau of Indian Standard. Now, according to them, has the following standard for a raw pasteurized milk. Okay, if we talk about the raw or pasteurized milk, then this will be the concentration of the microorganism. If we talk about the total plate count, okay, so if it is below 4 lakh, then it will come under a good category. Okay, if it is between the 4 lakh to 2 lakh, then it will be called a good. Okay, and if sorry, 2 lakh, uh, okay, if the range between the 20 lakh to 4 lakh, then it will be come under the category of good. And if it between the range of 20 lakh to 1 crore, then it should be fair. And if the total plate count is more than 1 crore, then it should be a poor category. Okay, coliform count per ml not more than 100 satisfactory. Okay, if the coliform more than 100, then it should be not satisfactory, means that it should not be consumed. So this is the microbiological standard for a raw cream. Now the next is your microbiological standard of a pasteurized cream. Okay, if we talk about the pasteurized cream, then the total plate count should not be exceed 30,000, and coliform should not be more than 1, and E. coli should be absent in 1 ml. Okay, then the microbiological standard in other countries have been laid down as follow. If we talk about the Canada, then the standard like count less than 50,000 per gram and coliform in there is no coliform in 1 gram, and test should be phosphatase test should be negative. Now, if we talk about the Northern Ireland, then untreated cream count less than 50,000 per gram. Pasteurized cream should not contain any coliform up to 10 gram. If we talk about the Sweden, then count should be less than 1 lakh per gram, and coliform less than 10 per gram, and aerobic spores less than 100 per gram. Okay, now the next is your plastic and frozen cream. The SPC count per ml should not be exceed 30,000. Yeast and mold not exceed 20 per ml. Means that if we take a 1 ml of cream, then the value of this yeast and mold should not more than 20, and coliform not more than 10 per ml or gram. The microbiological standard in other countries, like if we talk about the Canada, then the standard count should not less count should be less than 50,000 per gram. Coliform should be absent in 1 gram, and phosphatase test should be negative. Northern Ireland, then untreated cream count should less than 50,000 per gram. Okay, and your pasteurized cream should not contain any coliform up to 10 gram. And if we talk about the Sweden, then the count should be less than 1 lakh per gram, and coliform should be less than 10 per gram. If we take a 1 gram of cream, then in that cream, in plastic or frozen cream, the coliform number should not be more than 10. Okay, aerobic spore should less than 100 per gram. Method of manufacturing. Either of the following two methods should be used for the cream manufacturing from the milk. Okay, this is your milk, and when we convert this milk into a cream, then we take two methods that discuss in uh, here: gravity separation method and centrifugal separation method. Okay, so gravity separation method, in this method, what should we do? We kept our milk in a standby condition, and we are not disturbing it. Okay, so without disturbing it, we put it milk into a standby condition, and then we obtain cream. Okay, this method is commonly used in a home method. Okay, now next is your centrifugal separation. This separation are done by a separator, and the principle behind these two processes that fat become a lighter than the skim milk portion gets separated when the cream is subjected to either gravitational or centrifugation force. Okay, so when either we do a gravitational force or either we do this centrifugal force, then this fat become a lighter than a skim milk portion, and that's why it is separated from the milk. Okay, now the next is your microbiological quality of cream. Okay, the micro environment of cream and milk are the same, except the different in the proportion of various constitute like fat, protein, and lactose. Okay, so this micro environment means that the microflora of the cream and milk are same, only there is a difference between the constitute proportion of various constitute like fat, protein, and lactose. As a result, initial microbiological quality of cream is almost parallel to that of milk, but in later stages cream present more problem than the milk because of longer storage and irradiate distribution pattern. Okay, microbial number, it is believed that the if other things are constant, cream keep better than milk. So how this appear to be doubtful. Bacteria being a static due to higher viscosity of the cream may form clumps, and this lead to a lower bacterial count in a microbiological testing. Okay, see what happened, this cream form this bacteria this cream have a high viscosity, okay, due to which bacteria form a clumps, and that's why that leads to a lower bacterial count when we perform a microbiological testing of this cream. However, the same would not affect the methylene blue test result because the metabolic activities are not restricted under these conditions in a creams. Now the next is your, it has been reported by Davis that initial good cream with low count will keep for 6 days at 5 degrees Celsius temperature. Okay, if we have a good type of cream, then we kept it for 6 days at 5 degrees Celsius. But if there is an initially poor cream with high count, then it gets sour, develop a coliform, but fail to give positive methylene blue test under these conditions. Okay. In previous study of retail cream by a Colanso et al. 1966, the count were found to be quite high, over 10 to the power 5 to 10 to the power 8 per gram. Apart from the cold storage, it affects the microbial number in the pasteurized cream has been found to be greatly influenced by the cleanliness or the processing plant. Cream process in poorly clean plant, which give a initial colony count at 32 degrees Celsius, which would have included most of the psychotrophic bacteria of about 10 to the power 4 per ml, which increased to about 10 to the power 7 per ml after 5 days of the cold storage. Okay. Now on the other hand, after improved cleaning and sanitization, the initial count were only 10 to the power 2 per ml, which increased to 10 to the power 5 ml during a storage for 7 days. Okay. If the cream are prepared in a poorly unhygienic condition, okay, then at that time in the initial count at 32 degrees Celsius, it should be 10 to the power 4 per ml, and it should be increases if the cream are stored 5 days and reaches to the 10 to the power 7. Okay. But if the cleaning and sanitization condition improve, then the initial count would be 10 to the power 2 per ml, which increases 10 to the power 5 per ml when it is stored for a 7 days. Now the next is your types of microflora of a cream. A variety of bacteria, yeast and mold have been frequently encountered in a cream. Okay, if we talk about the bacteria, then bacteria flora cream comprises of the original flora of milk and the subsequent contaminant including pathogens. Okay, so this bacterial count are very similar to the bacterial count of the milk. The original flora mainly consists of micrococci, streptococci, lactic and non-lactic. This is your Corynebacteria, spore formers, aerobic and non-aerobic. Okay, and the contaminating organism entering cream during a manufacturing, it should be a gram-negative rods. Okay, this type of bacteria are come from the original flora, means that from the milk. But when we there are some contamination of the microorganisms will also enter into a cream when the cream are manufactured. Okay, then it should be it may be gram negative bacteria, staphylococci bacteria and lactobacilli. Okay, some pathogenic microorganisms also derive from a dairy personal during a processing, like your staphylococci, salmonella and other fecal type of other fecal type have also been reported in a cream. Now the next is your the predominance of microorganism in fresh cream totally depend upon the level of hygiene and temperature at which it is kept. Okay, see, we all know that this microorganisms are totally depend upon two conditions. One is your the condition, the level of the hygiene and the temperature at which we store. So here the cream, the predominant microorganism in a fresh cream will also depend upon two conditions. One is your the level of the hygiene and the temperature at which it is kept. The microflora of a cream processed in an unclean plant may include a very high number of psychrotrophs. If cream is not cooled repeatedly under the microorganism like Staphylococci, Lactobacilli, Bacillus cereus have generally been found to a predominant. Okay, that's why we have to cool the cream repeatedly. If we don't do is, then this type of microorganisms will predominant, like Staphylococci, Lactobacilli, Bacillus cereus. Okay, now on the other hand, prolonged storage at a low temperature also lead to us some predominant psychotrophic or psychotrophiles, mainly Pseudomonas, Okay, so if the storage time will increases, but storage time increases in a low temperature, then at that condition, or at that condition, there are some bacteria which are predominant, like your psychotrophs or and psychotrophiles, like your pseudomonas. Okay, gram negative, non-forming bacteria, oxidase positive, catalase positive, which enter the product as a contaminate from a dirty water. The psychotroph and the psychotrophiles encountered in a cream usually comprise of proteolytic and lipolytic type. Okay, the study on these lines indicate that the holding of the cream at 5 degrees Celsius and 30 degrees Celsius result in the predominance of microflora in the following order: at 5 degrees Celsius, Pseudomonas, especially non-fluorescent type, Alkaligenes, Acinetobacter, Acromobacter at 30 degrees Celsius. See, at 5 degrees Celsius, Pseudomonas, Alkaligenes, Acinetobacter, Acromobacter can grow. Okay, on and 30 degrees Celsius, Corynebacterium, Bacillus, Micrococcus, Lactobacillus, and Staphylococcus can be grow. The occurrence of psychotrophic coliform in refrigerated cream has been highlighted by several studies. Such coliform multiply slowly in cream during a refrigerated condition, that is the temperature between 3 to 5 degrees Celsius, and it increases 100 to 1000 fold in a few days. The composition of the psychotrophic microflora in refrigerated cream. Okay, the predominant of microorganisms in heat treated cream is totally depend upon the type of the heat. Okay, if we talk about the UHT process, then spore forming bacteria predominant in fresh pasteurized, sterilized or UHT processed food cream. Okay, this spore forming bacteria will be predominant in a fresh pasteurized or sterilized or UHT processed cream. Now, in canned cream, anaerobic spore forming bacteria may be predominant, and although these organisms are rarely of any important in cream. Thermodynamic organism which survive pasteurization temperature but not grow at these temperature. They are what? They will survive. Okay, but they cannot increase their number. Okay, now like your Streptococcus faecalis, they are associated species like Sarcina, Microbacterium, Micrococcus, and spore forming have been found to occur in a cream. Okay, now the next is your yeast and mold. Okay, so here the presence of yeast and molds are also reported in a cream. Okay, so if we talk about the yeast species, then it will be a Torola ceromoris, Candida pseudotropicalis, Trolopesis spherica, Rhodotrola mucilaginosa. And if we talk about the mold species, then it should be a Geotrichum candidum. Okay, now the next is your factor which affecting the microbiological quality of the milk. Okay, so there are various factors which affected the microbiological quality of the cream, and these factors are totally similar to the factor which affected the microbiological quality of the milk. However, the quality of the cream is complicated by prolonged storage requirement. The first one is your quality of the raw milk. The microbiological quality of the raw milk used for the separation has direct bearing on the quality of the cream obtained. Okay, if we take a poor quality of milk, then it will also give a poor quality of the cream, which will have a high another thermodynamic count, result in a poor keeping quality. Since these organisms survive the subsequent heat treatment of cream, the presence of Bacillus species type like Bacillus cereus, which lead of the cream in methylene blue reduction on MBR test. Okay, Raw milk quality. Firstly, the unhygienic production of milk at the farm level is therefore of great importance for obtaining good quality cream. Okay, if the milking are done in a hygienic condition, then obviously it will give a good quality of milk, and it will also give a good quality of cream. Secondly, the raw milk to be used for cream separation should not be stored for long at higher temperature. Okay, suppose that we have a milk and we store it at a higher temperature for a longer time, then again it will give a poor quality of the cream. The holding of the milk at low temperature may avoid souring, but might increase the psychrophilic and psychotrophic count. Okay, suppose that what should we do? We hold this milk at a low temperature. Then obviously it will avoid the souring but increases the psychotrophic and psychrophilic count. Although these organisms get killed during subsequent heat treatment of cream, their heat stable enzyme may survive, thereby which leading to a proteolytic and lipolytic spoilage of cream. The temperature of a milk should never be allowed to rise above a 5 degree Celsius in order to keep the growth of these organisms to a minimum. Okay, now the separation process will also affect the quality of the milk. In ordinary centrifugation separator, the temperature generally used for separating cream is between 35 to 45 degree Celsius, is ideal for the bacteria proliferation. Hence, the modern equipment should be used where low temperature, that is 25 to 30 degrees Celsius, are achievable. Okay, if the temperature are between 35 to 45 degrees Celsius, then it should be ideal for the growth of the bacteria. So that's why in a modern certification there is a temperature range should be 25 to 30 degrees Celsius. The use of lower temperature would also cause less physical damage to the fat globule. During separation process, the dirt, somatic or body cells and other foreign particles are removed as a slim from milk. The clumps of some bacteria like spore formers are also removed along with. In cream, obtaining a high spore count over 100 to per gram, the number of spore can be reduced by applying the high speed of centrifugal method, since the aerobic spore formers tends to form a chain. Now the next is the cream separated on the farm is expected to have a poor microbiological quality than the separated in dairy due to the relatively more unhygienic practices at the farm level. However, gap is diminishing with the advancement of the processing technology. The hygiene of the separator used for manufacturing cream therefore play an important role. The unclean separator might add the contaminant to the cream. After every run, the residue milk is constituted in the bowl, and between the disc must be carefully removed, as this would otherwise let microorganism proliferated and contaminate the subsequent batches of the cream. The cleaning with brush, draining the sanitization of the separator part is advisable. So this is all about the microbiology of the cream. I hope you will understand. If you have any queries related to this topic, you can ask me in a comment box, or for a further detail you can follow the ITLS Academy on different platforms like Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. Or for further query, you can ask your query to the helpline number that are already mentioned in your slide. Visit our website www.itlsacademy.com. Thank you for watching. Please like, share, and subscribe.